Hi everyone, today I would like to discuss the use of next-gen sequencing to study the molecular epidemiology of Carbapenem-resistant Acinetobacter baumannii. So Acinetobacter baumannii is naturally found in water and soil and typically is a commensal organism. However, it is frequently associated with nosocomial infections. Crabs, on the other hand, are recognized as increasing threat, and they have been multi-drug resistant before characterized as carbapenem resistant, and this is because carbapenem was the first line treatment for these multi-drug resistant organisms. So, like other nosocomial pathogens, crabs tend to affect those on ventilators, with indwelling devices, surgeries, prolonged hospital and ICU visits. And the organisms are typically transmitted through any contact with contaminated surfaces or the hands of infected individuals, and it affects the urinary tract, the lungs, wounds, and or blood. So as crabs are becoming more of an issue, scientists are trying to understand the various aspects of these bacteria to hopefully decrease the burden. There are several resistance mechanisms that have already been established, but despite this, the pathogenesis of this organism is not really understood very well. This is one of the reasons for the study that we will be discussing today. This study is unique as it is the first of its kind to look at the molecular epidemiology, distribution, and phylogenetic relationships of crab clones isolated from patients in the ICU. So over 6 years, the authors collected these samples and then utilized next-gen sequencing to hopefully obtain valuable insight on crabs. So let's briefly talk about next-gen sequencing. This method of DNA sequencing operates at a wide range. It is able to sequence both entire genomes and specific areas of study. This has already been used in multiple different research areas and has various uses. It has been seen in areas of research such as clinical genetics, microbiology, and oncology. So in this particular study, the specific system utilized is the aluminum HiSeq 2500 sequencing system. This system has been highly praised as there are several advantages linked to it. It is shown to have high throughput and rapid results. A wide range of sample sizes can be run. You can run 2 flow cells at a time. It has a high sensitivity and is very user-friendly. So in this particular study, sequencing the obtained samples were achieved by utilizing different preparation kits that are compatible with the HiSeq system. So first, DNA extraction was done utilizing the high pure bacterial DNA kit. The library prep was done using VATS prep kit for Illumina. The sequencing was done by the Illumina HiSeq 2500 system to obtain 2 by 150 base pair reads. And then data analysis occurred. So quality went through FastQC, the reads went through CLC Genomics Workbench, and genome annotation went through PGAP. And once sequenced and analyzed, what was found? So over the 6-year study, 302 strains were isolated and 105 were resistant to meropenem and imipenem. Of the isolated strains, 53.3% were isolated from those 65-year-old or greater. The plot that I've included illustrates the spatiotemporal, demographic, and clinical characteristics of the 105 resistant strains. Based off of a pangenomic analysis, single lineages seem to be dominant, and this was comprised of clusters 1, 2, and 3. Of the 9,506 genes that were clustered, 3,202 were homologous to all genomes included in the dataset. In regard to which clones were dominant, clusters 1 through 3 were identified. Each cluster is made up of clones based off of the relationships between SNP distance and the clustering on the generated phylogenetic trees. So both of the figures that I included gives us a good visual of this, and I think that the top figure is of utmost importance, in my opinion, as it shows time, age, gender, sample source, and clonality in regard to the isolate's phylogenies. So using a chi-squared test, the authors identified significant groupings on an epidemiologic level, as illustrated in this figure. There's a significant difference in community structure in relation to only time, not to any other of the factors. Over time, clone 1 was the most persistent, followed by 2, although 2 showed a gradual decrease in distribution over time. In regard to population growth, a DNASP analysis revealed that cluster 1 deviated from the expected value, but population expansion was present. After susceptibility was determined, it revealed all isolates demonstrated resistance to at least 3 antibiotic classes. The whole genome analysis further identified a combined total of 17 unique antibiotic-resistant genes, 55 unique insertion sequences, and 51 unique virulence factors. So what does all that mean? The data actually suggests that those who are elderly have a diminished immune system or in the ICU for a long amount of time have a heightened risk for developing disease. So crabs can cause a wide variety of nosocomial infections. However, the most common seen in this study affected the pulmonary system, where patients experienced pneumonia and respiratory failure. With that in mind, how did these patients acquire these infections in the first place? So it was found that in open wards such as post-op areas, increased potential cross-infection between patients and the overall inoculation of the organism. As discussed before, several medical devices and general hospital environment also contribute and can pose as sources of infection. In regard to resistance, the major genes that were implicated in carbapenem resistance were the BLA-OX23 and the BLA-OXA66. Something that was notable is many of the strains were tigecycline resistant, which is a non-carbapenem compound. So the virulence genes identified were those known to be responsible for the production and maintenance of biofilms. This is a major virulence factor in crabs. Outbreak situations were also observed in this 6-year study, which was supported by the fact that all the isolates belong to the ST2 clonal type. So this is of importance as ST2 is widely distributed throughout numerous countries and tend to form biofilms in larger quantities than that of the other strains. So of these clones, it was determined that all strains were dominated by a single lineage, and this was comprised of 3 clusters and 8 cones, which was a little bit different than what other studies have suggested. So of the 3, the dominant cluster seemed to change as time increased, which led to the discovery that the distribution of clone types were also changing. During this time of the most significant change, this is when the hospital that the study was being done at moved locations. However, clone 1 bounced back after the move, and it was dominant as of almost a year after the move. So this is thought that outbreaks can also be contributed to medical staff, as it was the same staff, and the equipment that they use, as one would think that the clones would not reappear upon relocation. So how is this study relevant? As you may think, there is a big concern of antimicrobial resistant organisms as of late. And this study is relevant as we should know more about these different organisms to ultimately maybe find prevention strategies so new organisms don't arise. And then this study can also eventually help in the development of alternative and novel treatment plans. And this kind of relates to possible future directions, as similar studies on different multi-drug resistant species can be done, and we can utilize this to generate sequences for virulence studies. So we have a lot of information from this study regarding the insertion sequences, virulence factors, antimicrobial resistant genes, and we can reduce the amount of crab cases that we see in the ICU.